Well, thank you for that introduction. Hopefully we'll go through and we'll have a smooth presentation here. Today I'm going to be talking about disease diagnosis and eco-friendly disease management. And because we have a lot of diverse people in the audience, I'm going to start with I want to provide you with the tools that will allow you to recognize, understand, and responsibly manage most of the diseases that you're going to encounter on shade and ornamental trees in the landscape. And because we have this diversity in the audience, we have some students here today, I'm going to start with some basics just to get everyone on the same page. And we can start with a basic definition of disease that you're probably all familiar with. Any condition that interferes with tree growth and development. And what's important to understand about disease, it's not a static thing, it's a continuous thing. It's an interaction between the tree host and the pathogen that's causing the disease. So you're going to see things are going to be dynamic, they're going to be ever-changing, they're going to be continuous, and they're going to be occurring over a period of time. And what's important about understanding disease as a living thing is to contrast that with an injury. And an injury is a one-time occurrence. It's something that happens to the tree. Things like frost damage or lightning. There's no interaction between the tree and that lightning. There's no interaction with the tree and the frost. Or if a car hits a tree or pulls it over. That's an injury. So we're going to be focusing on disease this afternoon. And when we're talking about disease, many of you are probably familiar with the disease triangle. But it's a good thing to start with because it emphasizes the important things that you need in order to have disease. First of all, you need to have a susceptible host plant. I guess my little arrow doesn't go that far. Yes, there was supposed to be a pointer here. Well, we can just... The left side, the host plant, you would need a susceptible host tree. Second, you need to have the causal agent. Third, the host and the causal agent need to interact in that bottom line, which is the environment. When all three conditions are met, you're going to see disease. So a good example would be scab on crabapple. Everyone's familiar with that disease. The host plant would be susceptible cultivars of crab. The causal agent would be the fungus Venturinia iniqualis. The environment would be one where it's fairly cool and wet in the spring as those leaves are emerging. And when you have all those conditions, you're going to see scab on the crab apples. Well, what happens if we select a resistant cultivar of crabapple? Red Jewel, for example, it's got some very good resistance. So you're selecting a genetically resistant cultivar. What happens? That arm of the triangle drops out. Even though the fungus is there, the weather is conducive for disease development, you're not going to see disease, or not going to have that level of scab that you would had you had a very susceptible cultivar. So we're going to come back to this idea of genetic resistance a little bit later in my presentation. Well, you've all seen the tree health problems. Tree health problems can basically be divided into two main categories. Abiotic problems that are associated with non-living factors, and these are problems that we can attribute to cultural or environmental factors. And these too, even though you're dealing with a cultural factor, it can be a cause that affects the tree over a period of time. With biotic factors, these are what most people associate with diseases. Living agents, bacteria, phytoplasmas, nematodes, viruses. As you know, out in nature, trees are affected by both abiotic and biotic problems. And a lot of times it's the old chicken and the egg scenario. Which came first? Did the abiotic problem weaken the tree and make it more vulnerable to the biotic problem, or vice versa? Well, it's well documented scientifically that abiotic problems frequently weaken trees and make them more vulnerable to the biotic problems. A good example would be drought stress. Many of us in the Northeast have had a very extensive drought this year, and we're going to be seeing some more biotic problems associated with that drought stress down the road. So I'm going to be focusing this afternoon on some of the biotic diseases. And when we're talking about diseases and disease prevention and management, we have two important things that we need to understand. We need to accurately diagnose the disease, and then we need to develop the appropriate tree health management program. And for many, disease diagnosis can be quite a daunting task. However, if we break it down into individual steps, a lot of times it's a lot easier to handle. Okay, so I'm going to go through briefly each of these steps this afternoon. And we're going to start with step 1, which seems like a no-brainer: identify the tree. And in most cases, all you need to do is identify the treated genus, but there are situations where you need to identify it to cultivar, or even to species, or even to cultivar, so that you know what those horticultural characteristics, or the characteristics of that tree. Is it supposed to have a variegated leaf, or are you dealing with some kind of phytotoxicity? So here you need to know what the normal attributes of that tree is, so that you can decide whether or not it's normal or abnormal. What should that tree look like at this time of year or at this particular stage of growth? Does it appear normal? Then you need to determine, if it doesn't appear normal, what's abnormal about it. And frequently we look at signs and symptoms in order to diagnose the disease. And in many cases, once you've identified the tree, it will give you immediately, it will tell you a certain number of diseases that may be common on that tree. Also, by looking at signs and symptoms, you can hone in, or you can make a short list of the potential problems that you might be dealing with. And symptoms are attributes of the tree itself, and they're the way the tree reacts, either externally or internally, to the presence of the pathogen. And there are many, many different types of symptoms, but to give you some examples, here we have rust on the left. In the middle we have a nectary canker, and on the right we have a gall. Those would be symptoms expressed by the tree as a result of infection. The other part of what we're looking for when we're trying to diagnose a tree is looking at signs. And signs are attributes or evidence of the pathogen or its products on the tree. And when you're looking for signs, a lot of times you'll be looking for things like bacterial ooze on the left on that developing crabapple with fire blight. You can see the bacteria oozing out of that developing fruit. In the center, there's a canker with fruiting structures of the fungus, Pycnidia. And on the right, you can see, obviously, this is a wood rot fungus fruiting a conch, or a bracket fungus fruiting on the side of an infected tree. So those would be signs. So by looking for signs and symptoms will give us frequently a good idea of what the causal agent might be. Also, we want to look at patterns and distribution of those abnormal trees. How many trees are affected? How many different species of trees? If it's all ashes, it might implicate one causal agent. But if you're dealing with ashes, elms, and maples, and shadblows all affected, you may be dealing with more of an environmental factor. Where are the trees located? Is there any pattern to the trees that are symptomatic? And this leads us to step 3, obtaining a really good and thorough background information and history on that tree. And sometimes it's actually asking the homeowner or your clients a lot of questions. For example, looking at weather patterns. You need to know if there's been an extended period of drought in that region, or alternatively, excessive rainfall. Is that tree subjected to prevailing winds or exposed to frosts? Other information that's important to gather about that tree would be site and soil characteristics. You need to have a good understanding of the soil composition. What's the pH? What are the macro and micronutrient levels in the soil? What's the soil texture? The bulk density? Are you dealing with a compaction issue? What about the soil volume? Is that tree in a soil volume that's way too small for the size of that tree? Looking at the air and drainage patterns, water drainage patterns in the site itself. Cultural practices. What's the history of watering, fertilizing, pesticide use? When was the tree planted? Was it a B&B tree, or was it container grown, or was it a box tree that was held for a long period of time? What were the planting practices that were used if you weren't the arborist who installed the tree? Other factors. A lot of times people neglect to say that they had construction not too long ago, so half of the root system of trees were cut off. What are the foot traffic patterns around the base of the tree? Any underplantings where root damage may have occurred, and now you're seeing a pathogen that's entered the tree through the roots. Where was the tree originally grown? Was it a southern tree brought up north? where it wasn't in the right province, or what other issues with air pollution. So getting a thorough background history on the tree can go a long way, especially when you're trying to diagnose some of those abiotic problems. So now where we've identified the tree, we've got a good history of the site and the background of that tree, the next is to try to identify the causal agent. And this can be done with experience, or alternatively, you can collect samples to submit to a specialist for identification. And I can't overemphasize the importance of accurate diagnosis, because if you're trying to control the wrong thing, any kind of management strategies aren't going to work because you've got the wrong target. So accurate diagnosis is important for two reasons: the need for control, or the need to do anything, and also the type of control or management program that you would initiate. And here, if you need help, this is where you really need to contact your local diagnostic laboratory. And in about the year 2000, the USDA provided funds to link all of the diagnostic laboratories in the United States together and created what they call the National Plant Diagnostic Network. So you can see that they divided the country into 5 regions. And here in Pennsylvania, as well as me in Connecticut, we're in the Northeast Division that you can see in the purple. But if you aren't sure if you have a diagnostic lab in your part of the country, you can go to this website and you can find your diagnostic laboratory. And they can help you with expertise, especially with the biotic pathogens. A lot of times we need to culture, we need to test for viruses, we need to do molecular diagnosis on your sample. And if you can contact those laboratories, they can help you quite a bit. We also have to put into this whole factor about what about those abiotic agents. Because we all know many trees can decline for something that's not a biotic agent, not a living agent. We can have mechanical injuries, chemical injuries, poor cultural practices, and physical issues that can impact the overall health of the tree. And this is, again, going back to getting that good site evaluation and good history on that tree, which is very important. So when we're talking about disease management, I like to put a more positive spin on it, not talking about managing the disease, but more so looking for a program for managing tree health. And essentially what I want to talk about is an integrated tree health management program. And here we're emphasizing the importance of tree health, because a lot of times healthy trees have the ability to withstand the challenges of disease or other stresses much more so than trees that are stressed right from the start. And this is where this whole eco-friendly component of my presentation this afternoon comes in. And we're going back to that disease triangle, because what I want to show you is when you break the pieces apart of the triangle, it will give you some idea of what you're doing in your tree health management program that has a direct impact on the disease itself. And we're going to start with efforts focusing on the pathogen component of the disease triangle. We use sanitation. We cut off limbs with cankers, or we rake some leaves away from the tree so that the source of the scab of the crabapple aren't going to be right under that tree. We use fungicides to reduce the populations of the pathogens that are going to be near or on or protect the tissue from that pathogen. So these are efforts aimed at the pathogen component of the disease triangle. We also look at the plant itself. We use resistant varieties or culture or species. And again, using that idea of genetic resistance. We optimize tree health, because as I mentioned, a vigorous tree is going to be able to withstand the challenges of disease much more so than a tree that's stressed. Then with the environment, we try to select the appropriate site for the tree. And then, or especially if we have recurring problems on a tree species with a lot of foliar diseases, you want to pay attention to the air drainage around that tree to promote the leaf drying. Also, we can use biological pesticides or biopesticides to create a hostile environment on the leaf surfaces predominantly. By adding a biopesticide, you're making that particular environment unfavorable for the pathogen, because we know we can't manipulate the real environment. We can't change the weather. We can't do anything about drought, we can't do anything about the weather, the weather patterns, but we can do things in the microenvironment of the tree. All those things come together and we have the bones or the basics of our tree health management program. And I just want to briefly go through some of the components of that before I start to talk about some of the specific diseases or key diseases. And when we're talking about disease management, the goal is not to completely eliminate diseases. That's just not possible, it's not realistic, and it's not necessary. We just want to keep them at manageable levels so that they're acceptable. They're not something that is objectionable. And we also have to realize that there are some diseases that are not going to kill the tree, whereas there are other diseases that we have to be more careful with that do have the potential to kill the tree. It's important we don't have zero tolerance for disease in landscape trees. Unlike if you're a nurseryman and you're growing these trees, you need to have a higher level of intolerance for disease because you want to produce healthy stock for your clients. So basically what we are going to be focusing on is prevention. We all know it's a lot easier to prevent a problem from developing than it is to try to pull out a full-blown disease epidemic on a tree or a number of trees. So we're going to start in first with cultural strategies, maintaining vigor, because this is really critical. As I mentioned, healthy, vigorous trees have the ability to compartmentalize, to wall off the ingress of pathogens. And this starts right from the start. This starts right from the start with tree selection and site selection, trying to select the appropriate tree for that particular site. Planting practices and spacing are very important, as are nutrient and water management. These are all aimed at maintaining a healthy tree. Mulches, we all know how important mulches are under the tree. Organic mulches help with soil moisture retention, they help with soil temperature moderation, and they do help with disease control by keeping up the general vigor of the tree, and they also help with remediation of soil compaction. Controlling weeds, especially for newly planted trees. You don't want the tree competing with weeds for available nutrients and water. Scouting. Most people don't think of disease as being scouting for disease. We always hear about scouting for insects and other and mites, but scouting is just as important for diseases. It allows you to identify or predict potential outbreaks. And this is where we have a job ahead of us to educate the clients, because they have to understand that your tree health management program might involve coming out to their property, regularly scheduled visits, assessing the trees, determining what's happening. If you're not out there and you're not putting on a fertilizer application, you're not putting on a spray of any kind, they feel like they're not getting their worth. And this is up to us to educate those people about the importance of scouting and watching out and recording for potential problems. It's very important. Okay, that leads us to the next major category, which is sanitation, selecting and planting pathogen-free stock, or certainly healthy trees that don't have major gouges out of them or potential cankers. I've seen some situations where a tree was just a poor specimen when it was purchased and installed. So you want to start out healthy. And in some cases, it's necessary to completely remove infected trees. If you have a heavily infected tree with bleeding canker, Phytophthora bleeding canker, for example, you may want to remove that tree if you have other potential hosts in the area. And certainly, pruning infected branches or limbs that have cankers will be very important and part of the sanitation process. And certainly, using clean equipment. Genetic resistance, another important piece of one of the tools in our toolbox for tree health management, using resistant or tolerant species or cultivars. And this is especially important for recurring diseases. And this is a very effective trait in terms of that's one whole arm of that disease triangle that you're manipulating. And unfortunately, it's not something that you can confer to trees that are already in the landscape. But if you have the opportunity to work with your clients, and you know you have recurring problems in the area, say you have recurring problems with rust or fire blight, on crabapple, you'd want to select varieties that have resistance or tolerance to those diseases. And that we are going to be seeing more and more, there's a lot of interest in breeding, and we're going to be seeing more and more available genetic resistance out there. We know we lived through this with the dogwood and dogwood anthracnose, where in the 80s, many of the flowering dogwoods in the eastern seaboard were wiped out as a result of anthracnose. We now have cultivars or crosses of dogwood with good resistance. As I mentioned with crabapple and scab, there's excellent horticultural characteristics on cultivars with resistance. Dutch elm disease, there are good cultivars with genetic resistance or tolerance to elm disease. The next category I want to talk about are the biological controls, which are also called biopesticides. And here we're actually talking about living organisms to control plant pathogenic organisms. And you can look at it very simplistically as the good guys versus the bad guys. Now, these biological pesticides have worked very, very well in the nursery and in the greenhouse, as well as the nursery industry. We're really not seeing that leap out into tree health care very much, but I have a feeling in the near future we're going to be seeing more and more effective biological controls for use in the tree industry. We have two modes of action. Some directly impact the pathogen itself by parasitizing the pathogen or antagonizing it. And then we have some indirect effects on some of the biopesticides where they actually increase or induce the resistance or the ability of the tree or enhance growth to be able to handle the pathogen. These are also, many people don't realize that biological controls are indeed biopesticides because they do have EPA numbers just like a traditional pesticide such as mancozeb would have. And some examples that are used would be the fungus trichoderma, and basically these are all most effective for the foliar pathogens. They have to be, trichoderma can be helpful for things like leaf spots and powdery mildews. Bacterium, Bacillus subtilis, which has a broad spectrum of activity against some of the leaf spots and mildews. And then we have verticillium, which is an isolate from potato that's used as an injection to protect elm trees from Dutch elm disease, as an example. And finally, chemical control. And you're probably wondering why I'm even talking about chemical control. This is supposed to be an eco-friendly presentation. Well, chemical controls, the ones that I'm going to be focusing on today's discussion, are going to be considered to be more of the products that are used with organic land care or with an integrated management program. And we have three main categories. We have the biological pesticides, we have the biorationals, and we also have what I would consider to be the traditional or chemical products. But the important thing, regardless of what kind of pesticide you're using, there are two important things to remember. What and when. When to treat. Most of the fungicides that we have available to us, especially the biopesticides, are protectants in their mode of action. Therefore, they need to be applied to the tissue, especially the leaves or the needles, before the infection occurs. So if you're going to be seeing symptoms on needles, or you're going to be seeing symptoms on leaves, they're usually too late for those applications to be even put on a fungicide application at that point. So you have to treat on a preventative basis, and very commonly for foliar pathogens, that means when those new leaves or those new needles are emerging in the spring, before you see symptoms. What to use? This goes back to that whole concept of accurate diagnosis. Knowing the target pathogen that you're trying to control. Because not all fungicides are created equal. We don't have all the same efficacies, especially if you're trying to control bacterium with a product that's only going to control certain fungi. So you really need to know what you're trying to control so you can select the most efficacious product. And this is especially true when you're using these soft pesticides as opposed to the more traditional products. We already talked about biopesticides. Biorationals, these are considered to be environmentally friendly products. Environmentally friendly because they have a minimal impact on non-target organisms and the environment. They also are considered to be more user-friendly. They have very high LD50s, so they're not only safer for you as the applicator to use, but a lot of homeowners feel happier and more content to have these products used on their property. And many of the biorational pesticides are OMRI approved, Organic Materials Review Institute, and many are acceptable for organic land care. And by and large, most of the biorational pesticides have limited efficacy, and that limited efficacy is usually focused on the foliar diseases. Examples would be the things like the potassium bicarbonates, the oils, the horticultural or neem oils, or some of the soaps. Then we have what I'm considering the traditional or chemical pesticides. And these are basically products with traditional modes of action. And some of these, the ones that I have listed here, many of them are acceptable for organic land care. Especially products, these are going back to the first fungicides that were ever developed or used. Things like copper products, sulfur, and some of the newer phosphorus acid products are acceptable for some eco-friendly landscape. They're not acceptable for organic land care. So we've talked about the importance of the steps in diagnosing diseases. We've talked about some of the steps in your tree health management program. Now I want to talk about some of the specific diseases that we encounter. And we all know in my 28, 29 plus years of being a plant pathologist, I don't know all diseases on all trees, and I'm still learning. We all still learn. I'm seeing new problems all the time, or new twists on problems. So it's not, you just can't, you're never going to know all the diseases on all the trees. But what I want to do is to help you to focus on learning to recognize and manage key diseases. And this will allow you to make informed decisions about most of the problems that you're going to encounter on both landscape, on trees that shade, as well as ornamental trees in the landscape. And the big thing here is to learn to recognize the types of disease, not the details. I'm not going to give you specific fungal names. I'm not even going to give you specific trees. I want you to focus on the symptoms that we're looking at so that you can sort of categorize when you go out to a property. You can say, okay, that's a foliar disease, or that's a root rot, or that's a canker, or that's a vascular wilt, rather than be worried about if that's a particular, the genus and species of the fungus or the causal agent. So when I talk about key diseases, I consider these to be diseases that are problems on many different types of trees every year. They're basically recurring diseases. And for today's presentation, I've divided them into 4 main categories. We're going to talk about foliar diseases, blight and dieback diseases, vascular diseases, and then stem and root diseases. And we're going to start in with foliar diseases because these are the diseases that everyone notices. Most clients, I deal with homeowners in our diagnostic office, many people get real bent out of shape about foliar diseases. But in fact, they're the diseases that have the least impact on the overall health of the tree. And I'm going to be dividing these in turn into leaf spots, anthracnoses, and needle casts, then the powdery mildews, and then the rusts, because they each have slightly different attributes in terms of recognition of that type of disease. Leaf spots basically can be characterized as spots of dead tissue scattered over the surface of the leaf. The size, the shape, the color can be variable depending on the host and the pathogen. And in some cases, in many cases, when we have cool, wet weather in the spring, you can have substantial and often disconcerting defoliation as a result of leaf spot diseases. Needle casts are similar to leaf spots, only they're on the conifers, where we have some yellowing or chlorosis of the needles, browning and shriveling, and premature casting or dropping of needles. And then anthracnoses. These appear often as more irregular necrotic areas on the leaves. A lot of times they're shaped by the venation pattern of the leaf. They can be spotty, they can be blotchy, they can be irregular, and in some cases they can cause a slight amount of twig dieback on the twig, on the new growth. So leaf spots, needle casts, and anthracnoses can be caused by fungi, bacteria, nematodes. They generally are host specific. So the leaf spot you may see on the red bud is not necessarily the same leaf spot that's infecting the maple. So they tend to be host specific. And most do require water on the free water on the surface of the tissue in order to infect. So as a consequence, these leaf spot and these foliar diseases tend to be much more problematic when we have wet weather in the spring, right when those tissues are breaking bud, when those needles or leaves are immature, which is when the primary infection is beginning. We talked about scab of crabapple earlier. Oftentimes you'll see the spots on the leaves. They can be olive and fuzzy. Sometimes they have like a feathery margin. And often you'll just see a lot of yellowing leaves. Will cause a lot of premature leaf drop, and you've seen highly susceptible crabapples in July with practically no leaves on them, but they come back the next year, full foliage. So year after year on an otherwise healthy tree, they can handle that defoliation. Tar spot of maple, problem that people usually notice when they're raking their leaves in the fall, but in fact you're looking at symptoms in the fall or late summer when the infection occurred in the spring. So when those leaves were emerging and breaking bud when there was free moisture. Septoria leaf spot of maple, particularly sugar maple, was a real problem the last couple of years in the Northeast, where we had significant defoliation and yellowing of sugar maples associated with these small necrotic spots that you see on the left, where all the spots coalesce into more angular lesions, necrotic lesions, and then would drop off the tree. This was an unusual outbreak. Black spot of elm, recurring problem, often causing a lot of chlorosis and defoliation. And this is one where I'm happy to see black spot come into our diagnostic laboratory, because usually people are bringing it in thinking they have Dutch elm disease. So it's a wonderful occurrence when you say you only have black spot, you don't have Dutch elm disease, even though it looks like you have a lot of chlorosis. But this is a foliar disease. It doesn't have any long-term implications for tree health. Tubacia leaf spot on oak, another problem we've been seeing in the Northeast the last couple of years, where we have these necrotic spots and that develop into blotches and cause premature drop or defoliation on oaks. And you're probably all familiar with oak leaf blister, and which develops into the blistered, and the trees get a generally off-colored appearance by the middle to the end of the summer. But you're dealing with infections that occurred on those immature leaves as they're emerging. A lot of problems on the conifers the last couple of years. Usually we think more, when I think of conifers, I usually think more of insect problems, mites or scales. But the last couple of years we've been seeing an upswing in the foliage diseases. And rhizosphere needle cast of spruce has been quite problematic. I think in our diagnostic lab we probably have looked at 40 or 50 spruce samples in the last 2 weeks from arborists. And you're seeing a lot of off-colored needles on current season's growth. And this is a disease that's much more problematic on spruces, especially drought-stressed spruce. And that's what we had in the Northeast this year. And if you have your hand lens out there on the top and the bottom right, you'll actually see the little signs, the fruiting structures of the rhizosphere needlecast fungus coming through those stomates. Another disease that was a real outbreak this year was Canevrigella needlecast of white pine. There were a lot of alerts coming from Canada all the way down the East Coast with this disease. And this is a disease, needlecast disease, that infects current season's needles. Infection occurs in the spring as those needles as they're emerging and breaking bud. And then the infection, the symptoms really don't show up until late summer, early fall, winter, or the following spring. And on the bottom right, you can see little shiny black sort of blister-like structures on the needles, and this is a fungus that's related to the tar spot fungus. So it was quite widespread throughout the Northeast, even in forest trees as well as landscape trees. And this is one that was discovered here in Pennsylvania in 1996, and I didn't see it in Connecticut until 1998. sort of been at a low level for many, many years, and it's just this year, because we had such a wet year in 2009, we started to see the problems in 2010, this spring. Another needle cast disease that's on the increase, rhabdocline needle cast of Douglas fir. This is a disease that I used to think was just a Christmas tree plantation disease, but now we're seeing more and more Doug firs planted in the landscape, and as a consequence, we're seeing more and more rhizosphere needle cast. Symptoms of infection, the infection occurs on the newly emerging needles in the spring. You don't have any evidence of infection until maybe the fall when you see some chlorotic spots on those needles. And then it's during the winter and the spring that they develop that green-brown banding pattern on current season's needles. And then if in the spring, as temperatures warm up, you flip the needles over and you can see the needles split along the rows of stomates, producing the spores for the next season's infection. Moving on to the anthracnoses, dogwood anthracnose, I mentioned this is more of a problem with regard to overall tree health than most other foliar diseases, because even though this is a foliar pathogen, the fungus can cause the spots on the leaves, but then it can move from the petiole down into the twig, where it can move into the twig, cause some cankering, and eventually, if left unchecked, can get to the main trunk, where it can cause substantial branch dieback, as well as kill trees. And you can see the tree on the right with a lot of dead branches. Maple anthracnose, and you can see that venation pattern on the left, where you have a lot of times the necrosis, rather than being spotty, will appear to be along the veins. Or in some cases, depending, we have two maples here. One, we have a typical maple anthracnose that's venal. The one on the right with the Japanese maple, we have more more necrotic spots and less angularity to those. Sycamore anthracnose, one that's probably familiar to anyone here who has any sycamores. Recurring problem year after year, and you can see that sort of the pattern of venation that determines the necrosis. And every year we have sycamores, when we have cool, wet weather in the spring, sycamores look miserable. But by June or by July, they have 80–90% of the canopy, and you'd never know they had a problem. And then they come back the next year, go through that stress in the spring, but then refoliate, which is just more evidence of these are not diseases that have long-term health implications. Oak anthracnose, again, you can see the angular patterns of necrosis on the leaves. Moving now to powdery mildews, which have slightly very different symptoms than the needle casts or anthracnoses. And you're all probably familiar with powdery mildews. And these look essentially the same on all the hosts. And sometimes you might even miss the early symptoms, where the leaf has a little bit of chlorosis or chlorotic or pale green spots on it. And then the fungus will then develop its usual powdery or sparse or gray appearance. Because some powdery mildews are very fluffy, others are more sparse. And these are all the powdery mildews are all fungi. And they are usually very specialized and host specific. So you may see powdery mildew on sycamore, but that's not the same powdery mildew that's infecting the elm, which is not the same powdery mildew that's infecting the lilac. What's important about powdery mildews that distinguishes them, even though these are fungi, and I said most fungi require free water in order to infect, these do not necessarily require water, and all they require was high relative humidity and moderate temperatures. When infections occur on new young leaves, you can get some distortion. You can see the white powdery growth on the sycamore leaves, but they're buckled because of the presence of the powdery mildew infection. Powdery mildew of oak, remember these are all pretty much look alike. Powdery mildew of elm, and then powdery mildew of dogwood, which has a very sparse mycelium or fungal growth, and often associated with it is that purpling or anthocyanins coming out on those leaves. The final of the foliar categories are the rusts, and early symptoms of rust infection usually go unnoticed. And when they're visible on the upper surface of the leaves, they're pale areas. But as the fungus infects the leaf more substantially, you'll get some blistering or brightly colored pustules developing on those leaves. And these are also caused by their fungi. And these are also highly specialized and do not trivially move from different host to host. And some rusts can go from the same host to the same host over and over, and then other rusts require more than one host in order to complete their life cycle. And you're probably all familiar with the gymnosporangium rust, such as cedar apple rust. It needs to spend half of its life on crab apples and apples, and the other half on junipers, particularly red cedars. So you have to have both hosts within a relatively close proximity in order to have disease occur. And here we have the symptoms of cedar apple rust on crab apple, where you get those brightly colored lesions. And the spores that are produced on these leaves can only infect juniper. They can't infect other crabs in the area. They can only go to juniper, where they produce an innocuous, inconspicuous gall for most of the year. And then come spring, the fungus starts to grow in those galls, and you see those spectacular gelatinous teleal horns develop. And in our diagnostic lab, we always know it's cedar apple rust or gymnosporangium rust time. We get our first call from a homeowner that says, overnight, my cedar tree is just covered, space aliens, creatures invaded it. And you know that it's time to start looking out for cedar apple rust or the other related rusts. Ash leaf rust can be another problem, which alternates between ash trees and marsh and cord grasses, where you get some spots on the leaflets you can see on the left, and then even some infections on the petiole itself. You can see those swellings and the brightly colored spores. Pine gall rust, this is one that's going from pine to pine, and you can see the galls and the brightly colored rust spores. And another conifer rust that has been real problematic the last couple of years in the Northeast is repeating spruce needle rust, chrysomyxa rust, where this is one that can go from spruce to spruce to spruce. And I don't know how many hundreds of samples of this rust we looked at this spring. The rust infects the newly emerging needles in the spring. You don't see any evidence of symptoms on those needles until the late fall or winter, when you see chlorotic bands or spots develop on the needles. And then come spring, they break open, and you can find all those rusty colored spores that are right on the one-year-old needles. They're right there to infect those newly emerging current season needles, causing a substantial amount of defoliation. Well, how do we manage these foliar diseases? Maintaining vigor is very important, and following sound cultural practices. Because if you're dealing with premature defoliation or premature casting of the needles, you need to keep up the vigor of the tree. Using good sanitation, getting rid of any of those fallen leaves when it's possible. If there are any branches or twigs that have died back as a result of anthracnose, doing a finesse pruning. Using resistant cultivars is a very effective means, especially if you have recurring diseases that are happening. And spacing the trees to get good air circulation, because if the faster the leaves dry or the needles dry, the much more reduced conditions you're going to have in that environmental component of the disease triangle. As far as fungicides, there are biologicals as well as biorationals that on a case-by-case basis can be effective. The potassium bicarbonates can be effective against some of the powdery mildews. But these are all done and need to be applied on a preventative basis. So now we're going to move from the realm of diseases that have really limited overall health implications to the tree and go on to some of the more serious diseases. And the first category are the blight and dieback diseases. And these appear as conspicuous death to the growing tips. A lot of times you'll see a shoot come out and wilt or brown or blacken. And these tend to be more severe when we have wet weather, and they're more severe on trees that are stressed. And in fact, if left unchecked, sometimes these can result in a tree death. Blight and dieback diseases can be caused by a variety of different pathogens, including bacteria, fungi, and fungus-like organisms or water molds. And these are often associated with tree stress or exacerbated by tree stress. And the first one is fire blight. This is a bacterial disease. We don't see it all that often. It's got a fairly broad host range with regard to rosaceous plants, things like Amelanchier, crabapple, for example, and apple, traditional domestic apple. And we don't have this as necessarily a recurring problem, but when it does occur, it can be quite damaging. And I've worked with orchardists and seen a whole orchard wiped out in 1 year as a result of fire blight. And you can see on the bottom left, you can see the typical shepherd's crook, or the new growth that's been infected and quickly blighted out. This bacterium can produce stem cankers. You can see that bleeding stem canker on your right. And on certain cultivars of apple and crabapple, it can actually become systemic in the tree, especially if the tree happens to be grafted on a highly susceptible rootstock. And you can get those real funky, ugly root cankers, and you can see the discoloration and the killing of the rootstock as opposed to the scion material on certain cultivars of apple and crabapple. Okay, bacterial blight of lilac can be a problem, especially when we have a couple of spring frosts on a lilac. The injury from the frost damage allows a good place, a way for the bacteria to actually get in, and you can see the killing of the tips. Okay, diplodia blight, we've got a wide host range, including some spruces, pines, Doug firs. We think of diplodia blight most commonly on 2 or 3 needled pines, and especially trees that are under drought stress. We used to call this diplodia tip blight. It's gone through a bunch of different names, but what usually happens is the fungus, when the tree is stressed, will start to infect those newly emerging shoots. And as a consequence, the shoots are killed before they emerge. So instead of 2 or 3 or 4 inch needles on the tree, you're going to have 1/2 inch or 1 inch needles. And these usually they are tan, as you can see in the tip. On a healthy tree, the tree usually has the capability of compartmentalizing or walling off or stopping the fungus before it gets into the 1-year-old, 2-year-old, and 3-year-old wood. But if you're dealing with a weak tree, especially one that's been under drought stress for a number of years, the tree doesn't have that capability to respond. So the fungus, rather than being stopped at that new shoot, goes into 1-year-old, 2-year-old, 3-year-old wood, can kill branches, and when it gets to the main trunk, has the potential to kill that tree. And you can see the resin on the dead branch there, and the short needle symptom on the left. And if you're out and you have your hand lens on infected needles, pull back the sheath, and you can see the little black-beaked fruiting structures on those infected tan needles. Willow blight, we're seeing more and more willow blight, a complex of several fungi causing a blighting of those newly emerging shoots in the spring. And on young trees, this can actually kill trees in a couple of years if left unchecked. So when we're dealing with blight and dieback diseases, avoidance of stress, whether you're talking about waterlogged soil or dry conditions, can be very, very important. And using resistant varieties when they're available is an important strategy. And certainly prudent pruning is very important. Getting those blighted shoots and dead tissues off of the tree so that they're not there to keep continually infecting. Fungicides, as far as blights and diebacks, are a little dicey. However, some evidence that copper sprays can reduce the epiphytic populations of bacteria, either causing twig blights or fire blight can be helpful. Moving on to the vascular diseases, and these are characterized as wilting or a stunting or yellowing of the foliage. Sometimes you'll see an otherwise healthy canopy, but then 1 flagged limb 1 year. The next year you'll see 2 or 3 flagged limbs. And that's an indication that you may have a vascular problem. In this case, we're dealing with pathogens that are actually growing in the vascular tissues, either the phloem or the xylem. And once they're in that tree, they usually become systemic in the tree, moving around just as the vascular tissues are systemic in the tree. The pathogens follow those and become systemic. And in many cases, once these trees are infected, they cannot be cured. and in many cases, and they can result in tree death. Now, the number of years that it's going to take for that vascular pathogen to kill the tree can be variable. Sometimes you'll have tree death in 1 or 2 years. Sometimes you'll have tree death in 30 years. So it's oftentimes very hard to predict. But it also is impacted by many factors, including the weather and the health of the tree. In vascular diseases, we're talking about causal agents such as that include fungi, nematodes, as well as phytoplasmas. And these, since they're in the vascular tissues, they can be transmitted through root grafts from tree to tree. And some of the vascular diseases can be moved by insect vectors. And the first one, the one of real historical importance, is Dutch elm disease. And if you look at the tree on the left, in that little circle, you'll see a flagged limb of some chlorosis, possibly indicating that it was a fairly new infection by bark beetles, which transmit this disease. But once that fungus is introduced into the xylem of the tree, it becomes systemic, and shortly thereafter you're going to see symptoms throughout the entire canopy as the fungus grows and multiplies in that xylem of the tree. So if you see that flagging, you see that decline, the wilting, and then you also see some discoloration in the wood, it's a good bet that you have Dutch elm disease. Because this is a vascular pathogen, so you're seeing some discoloration in the wood as a result of the tree's reaction to the presence of the pathogen. But if you see that canopy symptom and you don't see a lot of vascular discoloration, don't dismiss it as not being Dutch elm disease, because a lot of times you will not see consistent vascular discoloration. In our laboratory, we routinely culture for Dutch elm disease. That's one of most of the arborists in the state will just ask for their clients, please culture this to confirm, yes, this is Dutch elm disease. As I mentioned, and this is one of my little cartoons, I wish it was this easy, because we do a lot of culturing for Dutch elm disease, which takes a lot more time than just a quick diagnosis. So we have the little Dutch shoes here. This is a fungus that's spread through root grafts of the xylem, but it's also spread through elm bark beetles. And you can see the holes and the feeding galleries where they acquire the pathogen on their bodies and then fly, and in the process of feeding, they infect. Okay, another problem, a vascular problem that has a very, very broad host range is the fungus verticillium. This is a pathogen that lives in the soil, and it enters the trees through the root system. And it can cause, as I said, it's got a very, very broad host range. And we've been seeing it problematically in our area on maples, especially the Japanese maples, especially when they're under drought stress, which we've had. This year we were in drought conditions. The last 2 years we were pretty wet, and then we went through a period of several years of very dry conditions. And we had a lot of Japanese maples die as a result of verticillium wilt. And it's particularly troublesome when you see it on a mature threadleaf maple, as you can see here. Again, we're seeing a wilting, sectoring of the tree, the left-hand portion showing chlorosis or flagging, the healthy canopy on the right. So a lot of times when you see those canopy symptoms and you see this vascular discoloration, that you can see this sort of olive green discoloration either in cross-section or longitudinal section on the left, it's a good bet that you have verticillium wilt, especially if you know what host you're dealing with and if it is a host to vert. However, we also do a lot of culturing for this organism for arborists in the state to confirm it, because we don't necessarily see a lot of consistent discoloration in the vascular tissues. Ash yellows. This is a phytoplasmal disease. It's a disease, a phytoplasm is similar to a bacterium. It has no cell wall. And here we're dealing with a pathogen that's not living in the xylem. This is a pathogen that's living in the phloem of the tree. And it's transmitted through root grafts and also through leaf hoppers. And this is a disease that's has gotten a lot more publicity in the last couple of years with the advent or the discovery or recurring new discoveries of emerald ash borer. because, as you know, with emerald ash borer, you can have a fairly quick decline of a tree, but with ash yellows, you can get very similar symptoms. And we also have ash decline, which is a combination of possibly this ash yellows and environmental factors. So I'm dealing with a lot of entomologists, and they're looking for emerald ash borer. And what we're finding is they're coming in. I didn't realize how much ash yellows there is out there, where you have just a general decline or thinning of the tree, and sometimes some witch's brooms on the main trunk. This is a phytoplasma that also will infect lilac and cause witch's brooming. Pine wilt nematode we've been seeing more and more of, especially on stressed trees. And this is a nematode that a plant parasitic nematode that's transmitted by pine sawyers. And you can see the insect on the top, and then in the middle photo is those little tubes are all nematodes that are lined up in the tissues of the sawyer. When they go on, they fly to the pine tree and will inoculate the tree with the nematode in the vascular system. Once the nematode gets in that tree, it's going to grow and multiply and cause a plugging of the xylem vessels. So management of vascular diseases using, again, our eco-friendly or our tree health management program. In some cases, pruning out, if you're dealing with Dutch elm disease, for example, and you have a new inoculation of the tree from a bark beetle, you see a small portion of the canopy showing symptoms, and the fungus hasn't become systemic in the tree, doing pruning of that limb, sometimes several feet below the symptoms, can remove the pathogen from the tree in early cases. But if you're dealing with something like verticillium wilt, it's coming up through the roots, pruning is not going to be all that helpful, but you may be knocking down some of the resident population of the fungus in the tree. Using resistant species or cultivars. I mentioned there are cultivars of Dutch elm disease, or with resistance to elm disease. Or if you have a problem with the verticillium wilt, that means the pathogen is in the soil. You're keeping up the resources that the tree has to compartmentalize and wall off these vascular pathogens as best it can. Avoiding stress and, in some cases, insect vector control is helpful. Okay, final category of diseases are the stem and root diseases. And again, we're still in that types of diseases that can represent substantial threat to tree, to the life of the tree. And here I'm going to just break them down into branch and stem canker diseases, and then we'll talk about some root diseases. Okay, with branch and stem cankers, these are infections that occur on the branches or even on the main trunk. They appear as definite areas that vary in color from the surrounding tissue. Sometimes you'll see swelling, sometimes you'll see sunken areas, you'll see target cankers, and everything beyond that canker tissue will then die back, and the branch will die. And then these can, once the cankers develop on the main trunk, you can potentially kill the tree. These are caused by fungi, fungus-like organisms, and bacteria, and they tend to be much more problematic on stressed trees. Cytospore canker has a fairly wide host range. We see it a lot in our area on blue spruces, Colorado spruces, especially on 60, 70, 80 foot specimen trees that have been growing in Connecticut or in the Northeast under, that's not their natural region. So their natural zone, they're Rocky Mountain species. So usually when they're growing that many years under stress, you start to see some flagging, some branch cankers developing. On the left, you can see them developing. And then on the right, because of the spruce bark, you don't see a real canker. But a lot of times, looking at resin flowing will give you a good indication that you have a canker developing. Nectary canker is showing up on a lot of stressed trees with a broad host range. You can see the cankered areas here, very distinct with the fruiting structures on them right now. Botryospheric canker, again, this is a disease that we tend to associate with stressed trees with a very broad host range. A fungal canker, and if you on the tree on the left, you can see the thinning of the canopy. If you follow that canopy back, you'll see some cankering on the branch or even the main trunk. Bleeding cankers have been very, very much in the arborist radar screen in our area, especially Phytophthora bleeding canker, and we've seen a lot of problems on the European beach. A number of different Phytophthora species causing bleeding cankers all over the main trunk, resulting in thinning and dieback of the canopy. Final, root diseases. With root diseases, you're going to see non-specific above ground symptoms. The trees are going to look like they're in decline, they're stressed, they may suddenly die. And what we're really dealing with is we need to look under the ground at the root crown area for actually what's happening. We get, as I mentioned, you can sometimes get a wilt, a leaf drop, a droop, dieback, and tree death. And these are caused by fungi, fungus-like organisms, and some bacteria. And again, you're going to hear me say this tree stress over, and you have heard me say it over and over, much more problematic. Okay, Phytophthora root rot. This is a fungus-like organism, used to be called a water mold, and it's got a very, very broad host range, which includes not only conifers, but deciduous trees as well. And you can see this non-specific dieback or decline of a tree on the left. And a lot of times, in order to diagnose this disease, you're not going to find any evidence of symptoms of the pathogen in the above ground portion of the tree. You're going to have to look down to the roots or the root crown area, where you will cut into the wood, and you'll see that distinctive cinnamon brown discoloration that we often associate with Phytophthora root rot. It's got a broad host range. Here we have some noble firs, which are important Christmas tree species out in the Pacific Northwest, where you have this general decline, a whole tree on the left, on the right, and some lower branch dieback beginning on the right. And in a grower field, frequently you'll see 1 or 2 trees dying back. But if you cut into the base of those trees, you will see those diagnostic symptoms, that cinnamon brown discoloration where there's the canker developing up along the base of the tree. And frequently with conifers, you'll see some resin flowing on the base of the tree on the left, but you cut into it and you'll be able to detect, you'll see that diagnostic discoloration. And a lot of times with Phytophthora root rot, this is a water mold. The pathogen has a motile swimming stage. So frequently, by looking for water patterns or drainage patterns, you'd be able to identify or at least get some clues as to how the pathogen is moving around. And in this example, this is a Christmas tree plantation. If we look at the top, we have a couple of infected trees at the top, and then we look at the water drainage pattern. You can see all those skips in that area as a result of the pathogen inoculating following the drainage pattern down the tree, down the slope. And this is also helpful even in less dramatic instances, but in properties where you have drainage issues or you know you have water issues, not only detecting a tree upstream, but looking for problems downstream in that drainage pattern will also be helpful. So, and this is where getting a good site analysis, looking at the site and understanding what's happening. Yes?
Why would you suppose it's petering out at the bottom of the slope?
Could be that by that time the pathogen's been trapped. These are living spores, motile spores. They're attracted to roots. They actually swim towards the roots. So it may be a dosage effect in that case. It's a good question. Okay, the final root pathogen I want to discuss this afternoon is Armillaria root rot, and this is another pathogen with a very broad host range, which includes many conifers as well as deciduous trees. And we can find it in all kinds of situations, in landscape situations or in orchard situations, where you'll have 1 or 2 trees just start to show some decline symptoms. And so you'll see a very similar general decline, non-specific above ground symptoms. And in this case, you see 1 arborvitae there that's dead and 2 that are in the state of decline. But if we looked at the other side, you could find a large old oak stump. What's that telling us? That this is a pathogen that can persist in dead stumps for up to 30 or 40 years. As long as the fungus has the right food source, You may not have any other trees around, but that fungus will sit there until you have some susceptible hosts. Then it'll move out and infect those trees. So again, the idea of doing a thorough site analysis is very important. And with conifers, it's non-specific above-ground symptoms again, but we often see, as with the Phytophthora root rot, we see that resin flow at the base of the tree. But if you cut it, unlike the situation with Phytophthora root rot, if you pulled this bark off or you cut into the bark, here you're going to actually see the fungal fruiting structures, which are the signs of the disease. And you'll see the white mycelial fans or fungal growth on the wood right under the bark, which is a good indication that there's armillaria root rot there. And this is a pathogen that is also called shoestring root rot. And sometimes you'll see the rhizomorphs or the structures of the fungus. We don't see these very often with conifers, but you will readily see them with some of the deciduous trees such as oaks. You'll see the rhizomorphs coming right off the bark and onto the leaf litter or on top of the mulch layer, moving towards other trees. The fungus is also moving in the ground, growing as mycelium from tree to tree. The other name for armillaria root rot is shoestring root rot, honey mushroom root rot. And if you're fortunate enough to be around a tree in the fall, you will see those fruiting structures coming up on the base of the tree. And on the slide, the photo on the left, you see that old tree stump, old oak in a conifer plantation, where you see the dead seedling or sapling there. Well, you have a perfect source of inoculum with those dead stumps left in the ground. And when I deal with Christmas tree growers, a lot of times if they're clearing virgin land and they say, yeah, I don't want to remove all those oak stumps, it's going to take that much longer to pull them, they'll leave them in. And then a couple of years later, after they've put in all their trees, they'll find trees starting to die, and there'll be a pattern. So you'll see a circular pattern, right, focusing back down to that stump that they didn't want to take the time to remove. So what do we do about managing stem and root diseases? Protecting the trees from stress, whether you're dealing with excess water problems with Phytophthora or drought problems, which is more problematic for armillaria root rot, is important. Planting resistant or tolerant species, maintaining overall vigor, and as I mentioned, sometimes removing the stump, the woody roots, will be very helpful because if you have armillaria root rot, because you're going to be removing the source of food for the pathogen. Fungicides certainly are not at all curative, regardless of the disease. Regardless of the stem or the root disease that you're dealing with. Biological controls, I have a lot of question marks here, and I voiced my opinion that these products don't have a lot of efficacy against these types of diseases. Sometimes coppers can be effective, as well as the phosphorus acids have shown some success with things like Phytophthora bleeding canker. So with that, I'd be happy to answer any questions that you might have. Yes? Okay, the question was going back to the Canevirgella needlecast of white pine. And she was asking how you manage this disease in a stand of pines. Is that right? There's no real need to do it. You know, this was the first year we've ever seen this explosion of the disease. We're going to be watching it. I've been working with some foresters in Canada where they were very concerned about their white pine forests. And we're going to be watching it to see what implications it has on tree health. To date, my only experience with Canevirgella has basically been working with Christmas tree growers on small trees, because that's where the problem was originally identified. We rarely have seen it to this extent on mature trees. So the jury's still out. We're still learning more about this disease. Yeah?
fed fire blight this year in West Virginia. I'm reading conflicting stories to wait until the fall to trim it. Reading other stories, trim it right away. What should we do?
Very good question. The question was about fire blight and whether you should prune as soon as you see the symptoms or wait until the fall or when it's dormant. And I agree with you. There are conflicting stories about the best time to prune. But I'm an advocate of sort of the ugly stub pruning method, which is one that actually was developed, I think it was down in Virginia, in Kearneysville. Alan Biggs was one of the people who was working on this, and they found that cutting back, getting that tissue off the tree as soon as you see it is better. And they call it ugly stub because you don't care where you're pruning. You prune back to 3-year-old, 2-year-old wood, and it's ugly. It's not a proper pruning cut, but you're doing it so then you will go back and then you'll do the good, proper pruning cut later on. But I'm an advocate, and others may have different experience, but it's better to get it off the tree as soon as you can, especially if you're dealing with, say, a domestic apple where you have a highly susceptible rootstock. That's the pathogen can go. It can move in 3 or 4 weeks, right down to the rootstock.
And prevention would be early in the spring.
Prevention
For fire blight?
For fire blight would be, copper sprays are frequently used to reduce the epiphytic population of the bacteria that are present on that tree. Because the antibiotics, for a landscape tree, I'm not a real advocate of using the antibiotics. They're only effective during bloom. Anyway. Yes?
In the case of verticillium and wilt, are other trees in the same yard or close proximity, are they in danger of getting it?
Okay.
If they are, how do you prevent it?
Okay, the question was on verticillium wilt. And if you know you have an infected tree in a yard and you have some other trees nearby, are they in danger of getting or becoming infected? Well, that's a good question, and you want to first know what species of trees are in the yard, because there are some trees that have a natural genetic resistance to the fungus. And a lot of times it's a question of where the hot spots might be, because this is a fungus that overwinters or over seasons. It can stay for a long time as these little tiny pepper grain structures called mycosclerotia that are pretty tough and resistant. So they can sit in the soil for many years. But what stimulates them is the root exudates coming from the trees. Stimulate them to grow, and then they'll infect the roots. So if you have a hot spot in the area of the yard where you have a high level of the fungus, may not necessarily be a reflection of other parts of the yard where you may not have, it's not uniformly distributed throughout the yard, is what I'm getting down to. So there's no way of really predicting, and there aren't any physical barriers that you can put in the ground, because it can spread through some root grafts, but it can also, you can't put a physical barrier in if you happen to have a couple of mycosclerotia around the root system of a tree that's farther away, another hot spot. It's a tough one to deal with. Yes? Well, the fungus has to get into the roots, and sometimes when you have shriveling or drying of those tender feeder roots from dry conditions or drought stress, it can provide additional infection courts for entrance into the tree. Okay. Physical. Oh, yeah. I guess I'm really not sure about the answer to that. If the trees, depending on when it's done, and if the tree's already been able to wall off those roots or compartmentalize down there. Yes?
How has the woolly adelgid and hemlock been treated recently, apart from horticultural oil?
Okay, the question was hemlock woolly adelgid treatment. Horticultural oil has been used as a biorational control. But we've had Rich Coles, one of my colleagues in Connecticut, has had excellent success with soil injections or soil applications of imidacloprid. He's found very good luck with that, as well as basil bark applications with safari too.
Is there a brand name?
For the imidacloprid,
Yeah.
Merit.
Merit.
Yeah.
Okay.
I mean, and homeowners are treating their trees. They buy the Bayer home tree insect control, and just soil applications can give season-long control. And Rich has found that sometimes it can give her 1 or even 2 years of control of the adelgid. Yes? It's not really a vascular disease, but the name is Discula destructiva. So with the name destructiva, it's causing canker. So it's not a vascular disease. It's actually killing the cambial tissues and moving in and causing killing cankers. It produces the foliar symptoms, but then it goes beyond from the petiole and can cause girdling twig cankers. Yes, it does produce some discoloration in the wood. There are a lot of diseases that aren't vascular pathogens that can discolor the wood. Yes?
How many of these are we moving around with our wood chips? Are we moving any disease around that way by giving away wood chips?
Okay, the question was about wood chips and how are you moving diseases around using them? Well, composted wood chips or seasoned wood chips, probably you're not moving a lot around. Fresh wood chips, you may be. There have been some studies. This jury's still out. I think we don't know enough. There was some work done in Minnesota where they were taking verticillium wilt infected trees, chipping the trees and applying those chips to susceptible trees, and they have found a higher incidence. But I don't really know, but if you know, and also depending on if it's a lot of tree diseases tend to, verticillium, armillaria, phytophthora, those have broad host ranges, but a lot of the diseases are more specific for that particular tree species. So by moving that infected material around, you may only be threatening one particular species. But I really don't, I don't know the answer to that. I don't think anyone knows the answer to that, but it's a real good concern considering how much biomass we have out there, and the demand for chips and the demand for mulch. As you know, the last 15 years, everyone has mulched beds. Everyone's using wood chips. Homeowners don't want to pay to have the trees taken away, so you chip them in place and then you let them deal with them. Yes?
It's going to destroy the insect that's in the ground and some of the insects that aid in root growth, or the bio...
part of the whole root rhizosphere.
Right. So if you were to, second part of the question, if you were to use the insecticide, Merit, in one part of the season, fall, and then in the spring, mid-spring, late spring, then use a mycorrhiza, will it affect the growth of the tree? You bring back new growth to the tips and stimulating the root system after killing the insect 6 months, 5 months prior.
Right, and that's a question. I don't know how much work has actually been done on the impact of imidacloprid on the mycorrhizal organisms.
Well, I've been doing it.
Okay.
For years, and it worked.
And you've had good success.
Great success.
Yeah.
One season on, merit, following season, as late as you can for the mycorrhiza, and it's producing trees. On a property we were just on 2 days ago. The difference between 3 trees in one area and areas that weren't treated.
So
provide the insecticide or the fertilization.
So you feel that the imidacloprid soil injection has been really screwing up the mycorrhizal relationships?
That it was by, I forget which pathologist did that, but what we tried to do was use it as an example or as an experiment 6 months, 5 months prior to using the nutrients to try to bring the system back. So you have to kill the insect first, but then you have to bring back the cluster.
Right.
Get new growth out and killing all the pathogens by using the insecticide. So we found it was a way of doing it and getting results on major problems.
Mm-hmm. Yeah.
Just something to throw out.
Yeah, no, it's interesting. I will actually, I'll ask Rich when we get it back what his impression is on that. I'm not an entomologist. I only know what he's used, so I really don't feel I can even address the real impact. Great. No, that would be great. We can have a conversation later. Okay, thank you.